Anyway, welcome everyone for the third day of the Technion Summer School on Computer Security, which this year is focusing on privacy. And our first speaker of the day is Mathias Humbert. Mathias is a graduate of EPFL at Polytechnique Fédérale Lausanne. I think we already have two of them yesterday, so that puts, I think, EPFL as the leading source of privacy in our summer school. After graduation, he became a data scientist in ETH and EPFL joint ventures now with the Swiss Army Research and Development, some cyber defense campus, which we actually hope we will hear a lot about. He's an expert in privacy preserving in the context of machine learning. He's won numerous awards. I think the most interesting one is recent one from NDSS, where he's worked on privacy of DNA, right?
Methylation, yeah.
Won the Distinguished Paper Award of NDSS, which is a very well-known and large paper. So, Mathias, the floor is yours.
Thanks for the nice introduction. How I need to share my screen? Okay. Desktop one, yes. Okay. Oh, sorry, I need to allow Zoom. Apparently, I need to give Zoom the access to my screen, which I thought was possible before. I did it before, but now they ask me to restart Zoom to be able to share my screen. Let me try again. Oh no, now it works. Okay. Okay, great. Up. So you see my screen?
Yes, we can see the screen.
Okay, great. Okay, see the slide as well. I don't know for the questions, if I can see them somewhere, if there are any.
So questions are requested to be put into the Q&A or into the chat, and we will give you interrupts when it will be.
Okay.
And of course, you can always say, now I'm open for questions, and we will have a discussion. Yeah. Okay?
Yeah, sure. Okay, great. So yeah, thanks for the invitation. Actually, it's a great honor to lecture at the Technion Summer School today. So yeah, as you said, I'm part now of the Cyber Defense Campus. I'm a senior researcher there. So the Cyber Defense Campus was created only 1 year ago by the Department of Defense, and we have offices both at ETH and at EPFL. There's already a video question. Okay. At least my presentation will be sent, yes, to answer the question. So yeah, we are co-located between EPFL, ETH, and our headquarters in the center, in the middle of Switzerland. So today I'm going to talk about machine learning for privacy and privacy for machine learning. So what do I mean with that? So first of all, I try to use machine learning to evaluate privacy risk at scales. Because how can we evaluate privacy in general? We evaluate privacy by mimicking the adversary's behavior. So that means that finding optimal inference attacks given the available data, the data available to the adversary. And so what is the best way to do that in an automated manner to infer this information at scale? It is machine learning nowadays, okay? Because we have a lot of data available out there. We'll see in the case of biomedical data that there are a lot of millions of records that are freely available, for instance, online. And this is very important to precisely quantify the risk and efficiently quantify the risks, privacy risks. Okay, so this is the attack part. Now, I also try to enhance privacy in machine learning settings, right? Because I think that in general, there is no one-size-fits-all solution. And if we want to have something that both at the same time optimize privacy, utility of the data, and efficiency, algorithmic efficiency. So if there are approaches such as differential privacy or cryptographic solutions that are very good in theory, but in some cases, in fact, in many cases, either you have to compromise on the utility part, or you have to compromise on the efficiency part. So in my work, I try to also take into account the context of the problem, such as the machine learning model that we are targeting, the machine learning phase also, whether this is the training phase or the inference test phase, also the data that we have to deal with, whether this is high dimensional data or the types of the data, that's binary or real values and so on, as we'll see next in this talk, and also the type of attack. So for instance, differential privacy assumes a very strong adversary with a lot of knowledge. But in general, I mean, the attack, the duality is a bit different. So we try to also, take into account kind of the attack that we want to protect against. So what I will present in this talk is also like defenses that are tailored to the attack that we'll cover essentially. Okay, so do not hesitate to interrupt me if you have any questions, by the way. So there are three key privacy criteria that have been defined by the Data Protection Working Party of the European Union back in 2014 already. So these are as follows. So first, there is the ability to single out someone, right? This is the ability to isolate, to uniquely identify some or all records of an individual in a data set, okay? So this is somehow the first step towards linkability. Linkability is the second main key privacy criterion identified by this working party. And linkability is the ability to link at least two records concerning the same individual or group of individuals, either in the same database or in two different databases. And if one of the database contains actual identifiers of the individual, then this can lead to re-identification, which is also known as de-anonymization. Okay. And finally, there is inference, which is a very generic term, which means the ability to deduce with significant probability the value of an attribute from the values of other attributes. Okay. So from these three privacy criteria, I will actually propose and cover here in this talk three key privacy attacks. So the first one is the linkability, what I just mentioned. Okay, I've removed singling out, because singling out in itself, it's not really a privacy attack. It's just the first step towards privacy, towards a privacy attack, which is the ability to uniquely identify some records. But then you need to have some other knowledge to re-identify or to make some real privacy impact. The second one is what I call attribute inference. This is what they call in the previous slide, what we've seen as inference, which is the ability to deduce the value of an attribute from the values of other attributes. And the third attack, which is quite new and which was not really recognized back in 2014, which is membership inference. So membership inference is the ability to deduce with significant probability that a specific record is part of a data set. And we'll see why this matters from a privacy perspective in a second. Okay. So to give you a more like a visual way of visual understanding of this different attack, I'll show you some examples with genomic sequences, because this will be the focus, the data focus of this talk. So if we have, for instance, a genomic sequence, but we have only half of it, we want to, for instance, infer the remaining part based on some correlations within the genome. Okay. We could also have access to the whole genome of someone and want to infer the genome of another person, and that's the first attack that I will show in this talk. Okay. Now we have also the linkability re-identification attacks that consist in linking records from, for instance, two databases, and this way. Okay. And now if we have some, some identifying information in one of the data set, then we can re-identify the first one, the data set on the left-hand side. And finally, we have the third attack, which is membership inference, where we have a data set on the right-hand side of, with many records. And we have somehow only black box access to this data set. So based only on the output XYZ of, showing some statistics about this data set, for instance, we want to understand, to infer whether, for instance, the genome that we have on the left-hand side is part of this data set. And this becomes privacy sensitive if the data set contains, for instance, only individuals carrying HIV, for instance. Then if we manage to infer that the owner of this genomic sequence has his genome on the data set, then we can also infer that he has or she has HIV with high probability. Okay. So in this talk, I will focus on biomedical data in particular, because this is very obviously privacy sensitive data, but also because it is more and more available. So this is essentially due to the quickly decreasing cost of molecular profiling in general and of genome sequencing in particular. So on this image, we can see that the cost of genome sequencing has dropped from $1 million back in 2001 per genome to less than $1,000 back in 2015 already. So as a consequence, the number of genomes that are sequenced every year is pretty much doubling every year. So if we follow this trend, and this was only until 2017, but if we follow this trend, we will reach 1 billion sequenced genome in 10 years, right? Actually, in less than 10 years now, because we are already in 2020. Okay, so there is a deluge of biomedical data that, of course, on the one hand, enables more data-driven and precise medicine, precision medicine, and personalized medicine, which is a great thing for healthcare. But on the other hand, means also that there is a lot of biomedical data that are available, not only on trusted databases, such as hospital databases, but also on public databases too. And we see also this trend of direct-to-consumer genetic testing services, such as 23andMe, that is also growing very fast, right? I mean, nowadays 23andMe or Ancestry DNA have tens of millions of customers already, meaning also tens of millions of genomes, or at least the most significant part of our genomes, which are called essentially SNPs. So then once you get, for instance, your genome sequenced by 23andMe, you can also freely share it on open platforms such as OpenSNP. Okay, so OpenSNP is a platform where you can where people can share their genomic data for instance, to help research. Okay, and there are other types of databases that are freely available, such as the Gene Expression Omnibus, that do not only contain genomic data, but also other types of biomedical data, such as epigenomic data or transcriptomic data. And we see that there were already millions of samples back in 2014, 15, and this trend is not going to slow down. So in this talk, I will cover different data of what I call the Human OSI Stack. If I may use a kind of a computer network analogy. So in in our human body, we have different layers in a sense. We have the genomic layer, which is the very bottom layer of the body, right? Physical layer, and up to the phenomic layer, where we have the phenotype, so meaning that how we look like, our physical abilities, also our diseases, for instance, what we really see in a sense. And in between, we have various types of biomedical elements and layers, such as a transcriptome that in a sense transcribe the information from the genome into the upper layers and into our cells and in the end our phenotypes. So in this talk, I will cover 3 different types of data, of biomedical data. The first one are genomic variants. So these are called SNPs, single nucleotide polymorphisms, and these are the most common variants in our genomes. So the data dimension there is about 200 million points. We have about 200 million SNPs in a sense. The data type is encoded at each position, at each SNP as 0, 1, and 2, because at each position we can be either a homozygous major, homozygous minor, or heterozygous, right? Based on the alleles that we inherit from our parents. And in terms of time evolution, our genome is very stable because our whole lifetime, so it doesn't change much except some mutations from time to time. Then I will cover also microRNA expression, which are of dimensions of much smaller dimension than the genome, that are genomic variants, which is we know about 5,000 microRNA expression points at the moment. However, the data type is in R, so it can be any real values, essentially most of the time positive real values, and it's varying a lot. Okay. And finally, we have DNA methylation, that is of data dimension in between the genomic variants and microRNA expression, and that is bounded between 0 and 1. Okay, it represents a percentage, a fraction of positions that are methylated in a given cell. So, and it's varying over time also, but not at all position and not as much as microRNA expression. microRNA expression, which can be influenced by our activities. For instance, if we practice sport, so from one day to another, it can change a lot. Okay, and obviously there are many health privacy risks. And actually, if we look at this at the US Breach portal, which is on the right-hand side here, this is hosted by the US Department of Health and Human Services. We have in total that there have been more than 2,000 data breaches since 2009, and here these are only the breaches that have been reported to the US Department of Health, right? Obviously, and these are breaches that involve at least 500 customers of patients. Okay, so this is a lot of attacks. So we know that, for instance, hackers are targeting more and more like hospitals and so on, also with ransomware, for instance. Now, it can be that also sensitive health data of thousands of patients end up online because of human mistakes, as reported in our Swiss local newspaper. It was a mistake by an employee of the Synlab group, a pharmaceutical group. And all this data available online can lead to discrimination, as very well portrayed by the movie Gattaca, that I encourage you to watch if you haven't done so before. And just to show you that this is not only science fiction, we know that nowadays there are many football, for instance, football teams that are sequencing the DNA of their players to better understand their physical predispositions. And this is even the case of our local football club here in Lausanne, which is not the best team in Switzerland, let's admit it, but still has the means and willingness to sequence the DNA of their players. And this was already quite a few years ago. And obviously, our very private medical data are also of interest for data miners and brokers that are now collecting a lot of these in order to make profit out of it, as reported in this the Guardian article here. Okay, and what even exacerbates the problem is that there exists interdependent privacy risk with biomedical and genomic data, right? So what is interdependent privacy? Interdependent privacy is when your privacy behavior affects others' privacy. This was the case, for instance, in the Facebook Cambridge Analytica, where the Cambridge Analytica application got access to the friends of the users who agreed to install this app. Okay, so there are inherent interdependent privacy risk with genomic data because of the correlations between family members, obviously, right? And this is especially the case in the extreme case where you have, for instance, identical twins, right, who share exactly the same genomic data. And the problem is that there is absolutely no legal protection framework to handle that, but there are still many open questions, such as the consent of the relatives if you decide to share your data online, or if you decide to get your genome sequenced by, for instance, direct-to-consumer genetic testing companies. So this requires proper risk assessment to be as informed as possible. And that's what we have done in this first work that we present here. And this is actually part of the attribute inference attacks. Okay, so now I want just to exemplify that this threat is actually real by taking a real world example. So we have this individual on OpenSNP who has decided to share his genomic data, its genotypes basically, that was sequenced by 23andMe. So we can see the download link there. Okay, with some other information such as physical characteristic. And this person actually also showed his real name that I've hidden here. But this real name actually allowed us to find this individual also online. It's on a famous social network, right? Where he was also showing some of his family members. So by combining these two channels of information, we can easily, in a sense, infer at least some of this family members' genomic information, right? So by sharing his genome, this person actually puts his relatives' genomic privacy at risk. So in this work, we wanted to precisely quantify clean genomic privacy risk by first designing efficient inference algorithms to run the reconstruction attacks given the available data, and then quantify this risk with respect to the amount of genomic data that is shared and the relatives sharing them, considering also the background knowledge available to the adversary. Okay, so how do we formalize that? So here on the right-hand side at the top, right, we see a matrix X, which represents all the genomic variants, all the SNPs of the whole N family members over M positions in total, right? And so here the adversary's objective is to compute the posterior marginal probabilities of the family SNPs, given that some of the SNPs are observed, for instance, for those of the relatives who decided to share their genomic data, and given also some priors on population statistics that we need. Okay, and we also make use in this attack the intergenome correlations, so basically family ties due to family ties, and the intragenome correlations, which is called linkage disequilibrium. Okay, so as we have seen, each element in the matrix can take 3 values, because it can be homozygous major, homozygous minor, or heterozygous. Okay, but the problem is that now, if we want to, with a naive approach, the marginalization has a computational complexity which is exponential in the number of variables in this matrix, right? Because you need to sum out all others, right? All of those variables. However, we have, thanks to the Mendelian inheritance law, we can cut, for instance, each joint probability of each column, okay, of variables, into smaller probabilities, either conditional probabilities or prior probabilities for the founders. So founders are all people in family trees who have no ancestors, at least no data for their ancestors, okay? Okay, and this is typically the shape of the structure, the basic structure of a Bayesian network. So Bayesian network is a graphical model that allows to represent random variables that are and conditional independence between these random variables. So here, the structure is clearly given by Mendelian inheritance law. That is that a child's genome is determined by his parents' genome, essentially, right? It's conditionally determined by his parents' genome. So he takes half of his information from the mother and half from the father, right? And picked uniformly at random. So we can easily model these conditional probabilities. Okay. Now, once we have this Bayesian network framework modeled, we can have some exact inference by using the belief propagation algorithm, which is also known as a sum product algorithm, because it actually pushes pushes the sum inside the products there of this huge joint probability that can be factorized, right? And this allows us to actually gain a lot of time in the inference time, right? Here we use even an algorithm that is a bit more specific, which is a junction tree algorithm, because we may have some loops, some indirect loops within the original Bayesian network due to siblings relationships. Okay. And in this case, we reach, if we consider that the columns, so each SNP position is independent from the other, we have a complexity which is linear in the number of SNPs and number of family members. Okay. Now, if we wanted to also take into account the correlations within a single genome, which exist, it's a bit more complicated. Here we have a sparse pairwise joint probability matrix, which represent these dependencies. And then we need to update the graphical model to a factor graph, for instance. Okay. So in general, there is a very good reference book that I encourage you to read. If you're interested in this topic of a probabilistic graphical model, this is a book written by Daphne Kohler from Stanford and Neil Friedman, who is in Israel, I think at the Hebrew University. So it's a very a deep book, actually, but it goes into all the details, and it's very interesting. So now that we have actually our inference, we have run our inference, we have some posterior probabilities of at each SNP and for each family members, right, I and j. Okay, so now based on that, we can, so the inference, the estimated value of the SNPs is represented as x at here. And then we can use different privacy metrics to quantify the genomic privacy of people or of individual SNPs in a sense. So either we can use the expected estimation error of the adversary if we have access to the ground truth, if we know actually the SNP of the targeted, the value of the targeted SNP, sorry. Or if we don't know it, then we can rely on some entropy metrics that measures the uncertainty of the adversary, okay, given the observed genome. So XO is the observed information, the observed SNPs. Or we can also use mutual information-based metric, which is also, which actually simplifies to also something that is, conditional entropy divided by the prior entropy. So here we evaluated our framework with real data, so a pedigree coming from Utah, containing four grandparents, 2 parents, and 5 children. We focused here in this on chromosome 1, which is the longest one, and it contains about 80,000 SNPs. And we relied on the 3 previously mentioned privacy metrics to quantify either genomic privacy or health privacy. So what we call genomic privacy is the average over all SNPs, or the privacy values over all SNPs. And what we call health privacy is, for instance, is related to a particular disease, okay? So in this case, we compute the weighted average over the SNPs and only over those SNPs that contribute to the considered disease. So if we represent this Utah family as with a Bayesian network model, we get this nice Bayesian network, right? With the 4 grandparents who are founders, so they have no ancestors. So we will use some prior probabilities for them if we don't observe the genome. And then we have the parents, P5 and P6, X5, X6, here that are conditionally determined by the grandparents, and the children were the same, determined by the parents. Okay, and now let's look at how parent P5's genomic privacy evolved when we gradually reveal his family member's genome. Okay, here focus on the SNPs on chromosome 1. So first, if we reveal, for instance, GP3, GP4, and P6, we have absolutely no impact on P5 privacy, right? They are still the prior privacy levels for all metrics, because obviously at this stage, these individuals are totally independent from a genomic point of view from P5's genome, from P5's data, right? Obviously. But now, if we observe one child of P5 and P6, so C7 here, we see that P5's genomic privacy drops by about 30%, to 40%, depending on the metric that we use, okay? And this is not only the effect of observing C7's data, but also now the effect also of P6, because once we observe C7's, somehow P5 and P6 data become conditionally dependent, right? So P6 data also influence and tells information about P5's data once we know at least one child. Okay, now if we reveal even more children, well, the privacy of P5 decreases down to about 20% of the original privacy of P5 when we were observing no other relatives. Okay, now if we observe also P5's parents, we drop to about 10% of the original of the prior privacy. But we see that they have not much impact at this stage because we have already observed a lot of genomic information from the family. Okay, just also I wanted to show you an example of the real attack that I mentioned at the beginning, right? At the beginning. So which consists in linking profiles from OpenSNP and Facebook. So there we managed to find 6 individuals who were sharing their genomic data on OpenSNP with the real identifiers and with the Facebook profiles with also containing also family members information. So in total, this was actually putting at risk the privacy of 29 relatives in these 6 different families. Here I focused on 2 families to show you some evaluation, regarding health privacy, and in particular regarding privacy of predisposition to Alzheimer's disease. So here for Alzheimer's disease, we focused on 2 SNPs that are equally contributing to the disease risk. And as by definition, we have only access to 1 person per family information. So we have only access to the information shared on OpenSNP by 1 individual for each family. And here we see the results, the evolution of privacy, in particular, in this case, entropy, because we don't have the ground truth for the attacked relatives, right? Because they have just Facebook profiles, but they don't have OpenSNP profiles. And we see, for instance, that for given the inferred, so the prior is at around 0.5 for the entropy value. And we see that for if the inferred relative on the x-axis is M or C, which is mother or child, then the health privacy related to Alzheimer's disease drops to 0.3, for instance, for family 1. So it drops by about 40% by just observing 1 person's genome, okay, 1 person's SNPs. Okay. So if there are no questions so far, I wanted to also show you, to finish on this first attribute inference attack, I wanted to advertise our interactive online tool for estimating your genomic privacy, which is available at this URL, santeperso.unil.ch. So it's kind of French URL, but it was designed to inform users, for instance, people who want to just, for instance, get their genome sequenced by 23andMe, for instance, or who want to know whether, if some of their relatives have been sequenced already, how their privacy evolves, right? And for that, we derived a single privacy score to be understandable to as many people as possible. And you can go on this website and build your own family tree, and then test also hypothetical scenarios, whether for instance, your father is sequenced or your son, and so on and so forth, and the impact on you or the impact on your relatives. It's kind of symmetrical in the sense you can test both scenarios. Okay, so just I wanted to show you that such machine learning models, the graphical models, can also be applied in other contexts, such as location privacy. And very briefly, actually, there are more and more collocation data available online, right? For instance, on social network, where you can tag people at a given place with you, or in a photo, for instance, you also can tag people, and this also represents some information that can improve adversaries' power to reduce users' location privacy. But the drawback of this information is that it decreases, obviously, the complexity of the inference attack for the adversary. So the traditional mobility model that was used so far is a hidden Markov model, right? It was used in the reference paper by Chokri et al. At S&P in 2011, right? But the problem is that this model did not really scale when users' location become to be correlated, right? So our approach is to extend this model to Bayesian networks and run also belief propagation on it. Okay, and this is not difficult in a sense because a hidden Markov model is just a special instance of a Bayesian network, of a Bayesian, of a Bayesian network, sorry. So if we take really a few toy examples, very simple example, where we have the mobility traces of 3 mobile users at 3 different points in time. So here we see in red the nodes representing the actual location, the random variables representing the actual location of the users, and in blue this is the obfuscated location instance. This is coming from the paper of Chokri et al. And actually, it was not so difficult to now include collocation information by just adding some auxiliary nodes in the networks, in the Bayesian network, showing the collocation between any pair of users. Okay. So now I'll move to the second main privacy attack, which is a linkability and re-identification. Okay. Here I will present two different works actually, and for each of them I will also present a defense, a tailored defense. Right. So far I haven't mentioned any defense for the first attack, which would be then obviously hiding the data in some way. But for these, I will present some defenses that we have designed. So in the first paper related to linkability, we have focused on MicroRNA expression. Okay, so I want just to give you some intuition about what MicroRNA expression data are. So basically, by showing you also the difference with the genomic, the genome. So basically, the genome contains a blueprint of what a cell can potentially do, whereas the MicroRNA expression regulates what a cell actually does. The genome is mostly fixed over time, as we have already discussed, whereas the MicroRNA expression can change a lot over time. And the genome can hint on risk of on predisposition of getting a diseases, whereas MicroRNA expression can tell whether you carry a disease at a given point in time. Okay, so in a sense, the genome would be our hardware, whereas the MicroRNA would be our software, yet another computer science analogy. So, but due to the high temporal variability of MicroRNA expression, the common belief was that there was no real privacy threat from microRNAs because it was too variable. Okay, so that's what we challenge in this work, is whether we can actually track MicroRNA expression profiles over time. And to do that, we have designed a linkability attack, a temporal linkability attack. So the idea of this attack is to match 2 data sets at T1 and T2. And the difference between T1 and T2 can be variable, as we'll see in our evaluation. So typically this would be leaked at T1, a database that has leaked, which includes some names, some re-identifiers. And the second one at T2 would be a public database, which is anonymized. Okay, and we want to know which sample from T1 correspond to which sample from T2. Okay, so in order to do that, we use some unsupervised learning approach to pre-process the data and reduce its dimensionality. So we have at the beginning, we have more than 1,000 microRNA points per sample. Okay, and we use a PCA, so principal component analysis, which is a method to do a dimensionality reduction, a quite traditional method. So here we have our TJ, so at time TJ of individual K, for instance, we input the vector into the PCA box, and then we get some vector projected on the PCA dimensions. Okay. So the advantage of this approach is that it has smaller dimensionality, obviously, it has unit variance, and it has uncorrelated components. Okay. So now, once we have these projected vectors, projected profiles, we have this problem of this typical problem of graph matching, right? And we want to find the assignment that minimizes, in a sense, the total distance between any pair of profiles that we have found. So in general, this you have a factorial n possible assignments. So this is a huge number, so you cannot basically go over it, right? If n is quite big, then it is intractable. But there are some existing algorithms to do graph matching in an efficient manner, such as the Hungarian algorithm or the Blossom algorithm. Okay, and with that, we can find the optimal assignment that minimizes the distance between matched profiles at T1 and T2 in a time complexity that is n to the 3. So here we have tested actually different distance to measure different distance metric to measure the distance between different profiles, microRNA profiles, and we found out that the Euclidean distance was the one performing the best. Okay, so now we have, of course, also tried this approach and evaluated this approach on 2 longitudinal data sets. So the first one was called the athletes dataset, was containing the data of 29 participants and 2 points in time over a time period of 1 week. So before and after exercising with no known disease. And we took the microRNAs both from blood cells and plasma cells. And the second one was, second data set was a lung cancer data set, so with 26 patients, and at this time, 8 different points in times over a time period of 18 months. So they were split by around 3 months. They were around 3 months between each time points, and all these participants were carrying lung cancer. However, in this case, we have taken the microRNA expression information only from the plasma cells. So let's look at some of the results. So this is on the x-axis. This is for the first athletes data sets. On the x-axis, you can see the number of PCA dimensions that we have considered, and we see that this is the accuracy of the matching is maximized at around a bit less than 40 PCA dimensions. We have a success rate of the matching of 90% with blood cells, and of around 48%, so of this value for plasma cells. And this can typically be explained by the fact that blood cells typically contain probably more information, are more stable also than plasma cells, which is essentially liquid, right? Which is made of water, essentially, most of it. And then we have the results for the other data sets, lung cancer data set, which corroborates actually the first results. Remember that these are only plasma cells microRNA. So we see that, yeah, the maximum success rate we reach is 55%, where the average success rate over any time shift is 29%. We didn't see a strong correlation actually between the time shift. We tested for various time shifts. We didn't see any significant correlation of the time shift on the results. So we see that we can reach a success up to 90% of linkability for blood-based samples, right? Now we also evaluated whether the success was changing a lot with the larger data sets, because we use actually quite small data sets. And we see that the success actually decreases very sharply with the blue line here with plasma cells, whereas it decreases mostly linearly for blood-based samples. So, voilà. So in order to improve the situation, here we have tested 2 defense mechanisms that are tailored to this attack, to this linkability attack. Okay, the first one is to hide non-relevant microRNA expressions, right? If we know them before, typically, because sometimes randomization is not an option for biomedical practitioners. They invest a lot of money to sequence, to profile this molecular data, right? And they don't want to add noise, typically, to the data afterwards. Okay, and this is the use case for this hiding mechanism is typically to make a diagnosis in a hospital, because you know already, you know, a priori what are the relevant microRNA expression to test for. One thing to take into account is there also exists correlation between microRNAs, like there exist correlations within the same, between genomic variants within the same genome. The second approach looks more like differential privacy in a sense, but on the raw data, which is where we added noise in a fully distributed and differentially private manner, providing what we defined as epigenome indistinguishability. And this is inspired by the work of Kostas Chatsikokakis at PETS, but also later at CCS, which they defined this geo indistinguishability in the context of location privacy. So there, in our case, the noise is drawn according to a multivariate Laplacian mechanism, so it follows what was also proposed for differential privacy in general for databases for output perturbation. Here it's really input perturbation, so we perturb the input data. And this defense mechanism would fit better for research purposes, for instance, for publishing a data set used in a study to enable to test new models and so on and so forth. So obviously there is always a trade-off between privacy and utility, as I said before. So here we will quantify the privacy as the ability to prevent linkability attacks, as we have seen it. And we will quantify the utility as the accuracy of the underlying binary classifier, right? So here we have a classifier, binary classifier between disease and healthy people based on their microRNA expression. And here we consider a radial support vector machine classifier. This is not coming out of nowhere. It's actually a classifier that was proposed in the literature, in biomedical science, and published and so on. So this is typically well founded in a sense. So now if we add noise to the microRNA expression, we can see that probably we may have some misclassification for some individuals, okay? So to evaluate the utility part, we made use of another data set containing more than 1,000 participants this time to be able also to run the machine learning, the SVM classifier, right? We need, of course, more data. These participants also had some diseases, one per participant essentially. And this is unfortunately at one time point. So we cannot have at the same time a longitudinal data set and many participants in the data set, at least not at the time when we did the study. So let's first look at how hiding microRNAs perform, okay? So here we see on the x-axis a decreasing number of microRNAs that are available for the attack, basically. And here we see the two curves of plasma and blood cells, blood microRNAs. So we see that both curves actually are quite robust to a decrease of microRNAs. We see that we have to reveal less than 100 microRNA to reach a success rate, at least for the blood-based microRNAs, smaller than 80%. Okay. Now let's look at the accuracy of the utility part, okay? For a given disease, we want to actually predict whether someone has multiple sclerosis, right? And we see that actually the baseline of accuracy without any protection mechanism, any privacy protection mechanism, is 99.2% with 40 microRNAs that correlate a lot with multiple sclerosis. Because this is in blue, the accuracy of the SVM. Now let's look at the matching attack, so the success of the adversary, the linkability attack, right? Here we see that it increases pretty fast with the number of microRNAs available, right? So we see that already with 40 microRNAs, we reach a success rate of about 90% already. So now we have to find a good trade-off between these two curves. We want to minimize the matching attack accuracy, and we want to maximize the accuracy of the SVM classifier. So for that case, for that specific disease, we can find kind of a good trade-off by revealing only 7 microRNA points, where the attack success decreases by 54%, and the SVM accuracy decreases only by 1% compared to the baseline. Now for glioma, we have another example, another trade-off with even fewer microRNAs released. In this case, we can decrease the attacker success by 80% with a decrease in SVM accuracy of only 1%. Now let's look at the second method, which is the probabilistic sanitization, randomization of the original data. Again, for the same disease, multiple sclerosis, okay? Here on the x-axis, now we have the epsilon. It is used to tune the noise that is added to the data. Okay, and this is increasing value of epsilon, meaning that decreasing value of privacy in a sense, as we can see with the red curve, which is the success of the adversary. With that approach, we can find a trade-off for a given value of epsilon, right, which is there. Okay, and we see that in this case, the privacy is, so epsilon here is 0.025, okay, fine. But then the privacy here is improved by 63%, so the attack success decreased by 63%, and the SVM accuracy drops by 0.65% only. Now, if you look at, for instance, this is tumor of stomach, we get even better results. So we get a attack success decreased by 70%, and SVM accuracy decreased by only 0.2%. Okay, so this is in a sense quite surprising that adding noise is better than hiding some microRNAs. Okay, but we have seen that actually, if we look here at the last bullet point, we see that noising actually provides a better utility privacy trade-off than hiding in general. So we have tested that for all 19 diseases and participants, and that we can double, in a sense, the privacy of participants at a utility cost that is more than 1% for the majority of diseases. Okay, so now there are no questions. I will move to the second attacks, the second linkability re-identification attack, this time against DNA methylation data. Okay. So I will go, there's a question. Okay. I will go rather quickly on this one.
Are there any questions? Please.
Okay.
Let's try again. So if there are any questions, write them in the Q&A. So it seems there are no questions. Let's continue.
Yeah, okay. So here, yeah, I will focus on the new data types, the DNA methylation data. So this is a very well understood epigenetic mechanisms, okay, that influence human health actually, or that is correlated with human health. So for instance, hypomethylation at a given point in the genome is associated with cancer. Smoking mother can influence child with asthma through these epigenetic mechanisms and so on. So this is very sensitive data, obviously, and its privacy must be protected. Okay. And so far, the current privacy practice by, for instance, researchers in the medical field is to publicly release the data, the DNA methylation data on databases such as the Gene Expression Omnibus, but to take two privacy precautions. The first privacy precaution is to obviously anonymize the samples, meaning removing all personal identifiers. And this is quite needed indeed. And the second privacy precaution, which is very important, is to not include any corresponding genomic data with this DNA methylation profile. That's the case because we know that the genome can be quite easily re-identified using various side channels. Okay, there is a now famous attack by Gimrek et al., published in Science in 2013, that was using actually some database available online on Y chromosome that was linking the Y chromosome to family names, and this allowed them to re-identify some of the genomes available online in an anonymized form. Then there is our attack that we presented at PETS in 2015, where we used, instead of using this kind of auxiliary database that was not actually online afterwards, we used a phenotypic trace, so visible, such as visible trace like eye color, hair color, and so on, to try to reconstruct the genome and the anonymized genomic databases. Okay, so this is, we know that the genome can be easily re-identified in a sense. So what we tried there in this work is to kind of, in fact, re-identify DNA methylation profile by matching them to the genomic variants, genomic profiles, basically. Okay, so we have on the left-hand side DNA methylation database, and on the right-hand side genomic database in the sense. So this could be here the gene expression on Libus, and on the right-hand side OpenSNP, with even it could be real identifiers on OpenSNP. Okay. So we're going to again run this linkability attack between now two different types of data, of biomedical data. And to do so, we use the kind of the Bayesian rule or Bayesian approach. Okay, so we want to know the probability that the given genome, given SNP, for instance, given belongs to it corresponds to a given methylation profile, DNA methylation profile, right? And actually, we can do that because there are some methylation points that are influenced by the genome, by the genomic variants. Some of them are influenced by the genomic variants. So we can easily learn actually this conditional probability of having some methylation values at some point, given the genomic variants, right? This can be learned on another data set typically, okay, in transfer very well. So now we have also, it's quite easy to know the prior on the distribution of genomic variants, the probability of G, okay? So we can then, using the Bayes rule, reconstruct, get the probability, the conditional probability of having a genome given the methylation, okay? And then we can sum over, we can multiply over all SNP position, over all methylation position, and get some kind of maximum likelihood estimator. And then the results are, I'm here summarizing the results of this study, and they are in fact quite impressive, because we have, in total, we have about 20 million different methylation regions. But by using only 293 methylation regions correlated with the genotype, obviously, we reach a matching accuracy between 97.5 and 100% against a genotype database, a genomic database of size greater than 2,500 participants. So compared to the previous attack against MicroRNA expression, where we had much fewer individuals, participants, this is very strong accuracy, very strong success power. So in this case, we said that, okay, maybe noising the data or hiding some of the DNA methylation position is not sufficient. So in this case, we may want to do something stronger in a sense, which is to use some cryptographic mechanism. So here we try to construct actually a private random forest classifier. And this is also something that has been proposed in the past, this random forest classifier for brain tumors, okay, to classify brain tumors, different subtypes of brain tumors. It was published in Clinical Epigenetics in 2015, okay? And what we added there is the private part, okay, the privacy part. And to do so, we use the homomorphic encryption scheme, so additively homomorphic encryption in particular, specifically. And we've shown that this was secured under honest but curious adversary model, meaning that both the machine learning provider does not learn the patient data, and the data owner, which could be the patient, cannot learn the machine learning model, because the machine learning model can be also part of some intellectual property and so on. And here the typical use case is in a clinical setting, it's not in a research setting, or a diagnosis performed by a third-party provider. Okay. Because in this case, what we have to understand is that the model, the random forest model, has been trained already. So we know already its parameter and so on, number of trees and so on. And we do only the inference part, okay, on encrypted data. Because training a random forest on encrypted data is another challenge, right? Okay. Maybe you'll see that in other lectures later in the summer school. So we implemented this random forest in C++. We based our classification on 900 methylation regions, classifying between 9 tumor subtypes, and with the original random forest model that contained 1,000 trees. Here I want to show you some results. So here you see the accuracy, the evolution of accuracy in red with respect to the number of trees that are used. So on the extreme right side, you have 1,000 trees, and on the left side, you have 1 tree. And we see that already with even less than 100 trees, we reach a very good accuracy already, almost as good as the accuracy of the original model with no privacy measures, right? And we see that with this number of trees, we have a computation that takes less than 2 hours. Still, it's a lot. It's a huge computational overhead, obviously, because otherwise you would take a few seconds probably without any encryption mechanisms. But still, it's kind of the best trade-off that we could find. If we had used 1,000 trees, it would have taken about 12 hours in total. Okay, so now I will move to the last big group of privacy attacks, which is membership inference, which is also the newest attack in a sense, the most trendy, I would say, at this time in the privacy and machine learning field. And so just to remind you, this is the ability to infer that a certain target is in a specific data set, okay? You have the target here, the genome, you want to know whether it's part of this huge data set, which can be a study on HIV patients, right? So this attack, as I said, is gaining momentum, and it's gaining momentum in academia in particular. So we have many awards for this topic. So we've, for instance, membership inference against location data, got the best paper at NDSS 2018. Membership inference against machine learning training data, which I will talk about at the end of this talk. Got the Caspar Boden Award, which is a very prestigious award in the privacy community. And the membership inference against DNA methylation beacons got the NDSS 2019 award, and this is one of my papers that you mentioned at the beginning. So here I will present you the attack against MicroRNA expression data, which was published in 2016 at CCS. So here we assume you have a database, a set T, of m microRNA expression of any individuals. And as we have seen, each microRNA point can be represented as a real value. So we have now a matrix of real values n times m. So this can be a study of association between microRNA and a disease, and this is as such, this is the information of membership in this database is sensitive. So now we release some summary statistics about a subset of the microRNAs. Okay. For instance, the average values of the microRNAs of the participants in this set T. And then we have the curse analyst, which is the attacker here, who has access to these summary statistics, but also to the actual microRNA expressions of the victim V, which is XV, which is the vector XV. And then her objective is to determine whether V is member of the pool T by using the summary statistics only about this pool, about this set, and XV. Now there are essentially two differences between of this membership inference attack between genomic data and microRNA expression data. The genomic membership inference against genomic data was proposed by Homer et al. Back in 2008, I think. And we are studying microRNA expression in this lecture, in this paper. So microRNA expression are basically real values, whereas as we have seen, genomic variants are ternary values. And also the dimensionality of microRNA expression profiles is order of magnitude smaller than the one of genomic data. Okay. So for this attack, we tested essentially two statistical tests. The first one was proposed by Humbert et al. In their original paper against genomic data, genomic variants. So here at each microRNA J, we compute actually the difference between the distance between the value of the victim XV and the mean expression in the reference population, and the distance between XV and the mean expression in the pool, in the set T that we want to attack, okay? So now if we look at it, if XV is not part of the pool, it should not influence mu at so much. So mu at should be more or less similar to mu in a sense, on average, right? And so this difference should tend to 0. However, if XV is part of the pool, it will somehow influence the mean expression in the pool, mu at. And as such, then this difference should be greater than 0. So now if we sum over all microRNA expressions that are released and that we know about the victim, we can then use a student t-test to determine whether V is part of the pool or not. Okay, and it's part of the pool if the sum is strictly greater than a threshold that we can tune. Now this L1 distance is okay, but there is even a better test, which is the likelihood ratio test, that provides actually the maximum achievable power for a given false positive level. This is due to the Neyman-Pearson lemma. Okay. And this likelihood ratio test can be represented as shown in the slides here. This is in fact the log likelihood ratio test. And we see that is sum over all microRNAs. And we see that actually if the standard deviation between the pool and the reference population is the same, when we have the log on the right-hand side that simplifies, is equal to 0. And then we see that we have, in fact, also two differences between Euclidean distance that are, in fact, normalized by the standard deviation of each microRNA expression point. Okay. So another advantage of the likelihood ratio test is that it allows us to find to derive a closed form relation between the power beta, so which is a true positive rate, the false positive rate alpha, okay, the number of exposed microRNA statistics, M, and the number of individuals in the pool, N. Okay, so what does this tell us in simple words? Okay, in simple words, we see with this formula that for a successful attack, we need the number of exposed microRNA statistics M to increase with the square of the individuals in the pool, of the individuals in the database. Okay, N. And if we compare with the previous work against genomic data, where it had to increase only linearly with N, it's a good result from a privacy perspective. Because if we manage to increase number of participants in databases, then the adversary needs to increase even more the number of microRNA points he has access to and microRNA statistics he has access to about the database. Okay. So we again evaluated this attack on real data coming from the gene expression omnibus. So actually the same data set that we used to evaluate the utility in the linkability attack, in the context of the linkability attack. So we have again more than 1,000 individuals with 90 diseases, and from 124 individuals with Wills tumor to 13 with stomach tumor. And we compute the reference population statistics over the whole set of individuals. Again, this is a data set that is publicly available in the GEO database. And we consider two different experimental settings there. We first construct the pool, the database, by randomly picking n individuals among the 1,000 plus individuals in the GEO dataset. And we construct also another database, which is disease specific, which could be typically a case group, right, in a research study, from n going to 13 for the stomach tumor patients to 124 for patients with Wills tumor. And we compare the two settings, right, in terms of attack success. So let's take first 30 individuals in the pool that are randomly selected from all individuals in the population. This is a ROC curve here that we see for different attacks, actually, LR, L1 distance, and the theoretical curve also. So we see the false positive rate on the x-axis and true positive rate on the y-axis, the power. Okay. Here, if we look at this point with a false positive rate of 10%, we have a true positive rate, a power of around 40% with its randomly selected individuals. Now we have the same size of the pool, 35 individuals, but this is a case group. Okay. And here we see that for the LR test, we have a slight decrease for this case group. So we have that, in this case, with this disease, we see that the results for randomly constructed pool and a pool that represents a case group with individuals sharing the same disease does not change much in this sense. Now let's look at the pool, a bigger pool, okay, of 124 individuals. We see that here the success is actually decreasing because we have only about 25% of true positive rate for the same false positive rate of 10% for the LR test. But then if we see for the same number of individuals but sharing all Wills tumor, we see a huge increase actually of the success, right? And why is that the case? Probably because the statistics now, the microRNA expressions are highly influenced by, highly perturbed in a sense by the Wills tumor, by the disease. So this gives some advantage to the adversary, okay, when he's running his membership inference. So here we have a true positive rate that jumps to 60% with the case group. So again, we tested to reduce the privacy risk. We tested two defense mechanisms. The first one is to release only a subset of microRNA expression statistics, so somehow hiding the statistics. So publish between 1 and 466 microRNA expression means. That is the maximum number we had access to. And the second mechanism is also based on differential privacies. It's a classical Laplace distribution scale to achieve epsilon differential privacy. So here the scale is the sensitivity of the average function, because we consider only this function, the statistics, the average, divided by epsilon. Okay, and the sensitivity of the average function is in our case equals to the sum over all microRNA expressions I that are of interest of the range of each microRNA expression divided by n. And actually the amount of noise to be added increases proportionally to the scale, to this sum, right, to the sensitivity of f, okay? And which is a bit problematic in our case because the range of each microRNA expression can be greater than 10,000 actually, because microRNA expression values are not bounded at all, and actually this will influence a lot the amount of noise that is added that has to be added to the statistics, and that will also damage utility quite a lot, but we'll see that. So first, let's look at the hiding mechanism, how it works. So this is the AUC on the y-axis, and on the x-axis we have the decreasing number of microRNAs. Okay, so the EUC, the greater the EUC, the better the inference actually. Yes, so yeah, EUC equals 0.5 means best privacy because this is like random guessing, and 1 means no privacy. So we see that already. This is also very robust to a decreasing number of released microRNA statistics. Right? We see that even for the curves starts to decrease seriously after around 100 or 80 microRNAs released. Okay, now let's look at the differentially private mechanisms, where we can here we can see the AUC curves in blue and green for different attacks, for two different approaches, and in red the noise to mean ratio curve for increased amount of noise. So the noise to mean ratio curve represents the utility in some sense. So 0 means all utility, and then if you have greater ratio, then you lose in utility, right? Okay. And you have increased noise. So we see that if we really want to decrease the attack performance, something that is like random guessing for both attacks, actually we see that the L1 distance attack is actually more robust to noise, in fact. Well, we need to have an epsilon that is such that the noise to mean ratio is of about 0.1. So it means that the amount of added noise represents 10% of the average value, essentially, which is quite huge. And this is quite a huge utility cost, in fact, with this data, with this particular microRNA data. Okay, so to summarize this study, we have seen that microRNA-based studies are prone to membership inference. However, the number of dimension M of number of microRNA statistics has to scale with the square of the number of individuals in the pool. And M also is much smaller than in genomic databases, as we have seen. Okay, so these two things are good from a privacy point of view. But the problem is that N, also number of participants in current microRNA database, is also much smaller because we have way fewer people whose microRNA expression is sequenced compared to the genomic people whose genomic data is sequenced. And also we have seen microRNA expression are way more affected by your health status, by your disease than the genotype, which is very stable. So it can give some more power to the adversary to make some inference, membership inference. So our high-level recommendation out of this study was to increase N in the pool, in the database, to at least 200 participants, individuals, which would mean that you have to have at least 40,000 microRNA statistics, which is not possible currently because there are not 40,000 microRNAs in total. Would be to be in a safe environment, and only add a bit of noise when really necessary. Okay, so we have seen this attack, membership inference attack, in the context of microRNA database. Okay, but there are other settings also where this has been performed. It has been performed against DNA methylation data as well. Okay, and this is a paper that will be actually presented tomorrow at USNP. Here also in this paper, we use actually also machine learning approach to find the most informative DNA methylation positions. There is some question maybe. Okay, no question. And also against location data, there's this paper at NDSS that got the best paper award in NDSS 2018. That targeted aggregate location data, and they use also some machine learning approach to optimize actually the data, the position that they used. And finally, there is the membership inference against training data set in machine learning settings, typically machine learning as a service. Okay. Now there is this paper by Chokri et al., published at SNP in 2017, membership inference attacks against machine learning models. And there is our paper, ML Leaks, published at NDSS 2009. And now I will present to finish this lecture, I will present this topic in more details. Okay, so this was the previous settings that we have seen, right? We have a database, for instance, of microRNA expression data and some summary statistics released about this microRNA expression, right? We have the chorus analyst who has access to the victim's data. So what changes with the machine learning setting? The machine learning setting, the attacker will actually query the machine learning as a service provider, such as Google Cloud or the Amazon, same AWS, with a sample with a record of the victim, this cat in this particular example. And then the machine learning as a service will return the prediction vector, the outcome, which is the prediction over all possible classes. And now the objective of the attacker is to determine whether V, the victim, is part of the training set of this machine learning as a service system by just using the output distribution that she now has access to over all possible classes. Okay. So in the state-of-the-art, this is state-of-the-art attack, which was proposed by Chokri et al. 2017. So we have the target data set of the machine learning as a service and target model. So the target data set is used, the training data set used to construct the target model. So how do we build some ground truths to construct the attack model? So to do so, we need to have to build shadow models that mimic the target model. Okay. And actually in their work, they needed a lot of shadow models. Okay. And to construct the shadow models, they need, of course, their own data set because they don't have access to the target data set, which is private, basically. They need their own data set, local data set, that needs to follow actually the same distribution of the target data set. So in order to build proper shadow models that are similar to the target model, okay? And to construct this local data set, to construct this local data, actually they are in fact querying a lot the target model to get this data, because then if the target model's prediction has a very low actually entropy and so on, then it means that probably the local data set that they have is correlated, is similar to the target data set. Okay, but this requires a lot of queries to the target model, which is costly. Okay, and then once they have this local data set, they split it into training and test set in a sense, so where some of the data are used to construct the shadow models and some are not used to construct the shadow models. And then they take the outcome of these shadow models, the distribution over possible classes, and input that into the attack models. Okay, the attack models are also machine learning models, but that do not classify whether you are a cat, a dog, or panda, but that does some binary classification whether you are in the training data set or not. And then at the end, when all these models are constructed, they make one query to the target model with the victim's data that returns the distribution of possible classes, and then we input this distribution in the target model that you have constructed to decide whether it's in or not. So the drawback of this first paper is that they have to construct a lot of shadow models in order to have high accuracy. And this is costly, obviously, because you need then to have a local data set. And this, you need to query the target model that is not for free. And for instance, target model could also block you if you make a lot of requests, a lot of queries to them. Then this means also they need also a lot of attack models, one per classes at least. And they assume also that the local data set has the same distribution as the target data set. Okay. And essentially they also assume that the shadow model has the same structure, has the same machine learning structure, machine learning model as the target data set, which is also a rather strong assumption. So in our work at the NDSS 2019 paper, we actually studied 3 different and more realistic attacks, in our opinion. 3 new adversary models. So first, we need to construct only one single shadow model with a different structure than the target model. So this means that the attack is less costly, obviously, because you need to make fewer queries to the machine learning as a service system. And let's look at the results now. Here we show the precision and recall for various data sets. So the classical, the usual suspect like C410, MNIST, purchase data set. Okay, and in blue you see the results of Chokri et al, and in green you see our results. Right? And we see that in most of the cases, actually our results almost outperform, at least for the precision, the approach of Chokri et al. And for the recall are pretty similar, right? Okay. Yeah. We outperform by a few percentage points in a sense. So we see that even with this less costly attack that can be also more difficult to detect by the machine learning as a service, we can still reach very good results, empirically at least. Then we consider another, even a more relaxed adversary model, which is with the same assumption as one, plus a different distribution of the local data set compared to the original training set, right? This is what we call data transferring attack or transfer learning, where we have a different data set than the training set. And here we see, so the precision recall again, where we have on the y-axis the training set, the original training set, and on the x-axis the training set, the local data set that we use to construct the shadow models. And we see that for some, actually, at least for some of the data set, we see that, for instance, if it was trained with news data set and it's the local data set is completely different, like an image data set like C410, we reach still quite good results, surprisingly. Or if you train with the image data set and then you have your local data set, which is news data set, then also it was the membership inference attack is quite powerful still. So the results in general, if we compare for all pairs of training set, local set, the results decrease by only a few %. Okay. And finally, we consider another last adversary model, which is without any shadow model, with a different distribution than the training set, with no training phase at all, but just we rely on the output distribution only. Okay, and from the output distribution, we compute different statistics, such as the maximum value or the entropy over the output distribution. And this happens to be essentially sufficient in some cases, at least for about half of the tested data sets. So now why is this attack possible? Why is this attack so powerful? It is essentially due to the fact that the training samples tend to overfit. Okay, so for the training samples, if you query the machine learning as a service with training samples, you'll have on average better results. In a sense, that's what overfitting means. And so to fight against this membership inference attack, instead of relying on classical privacy solutions like differential privacy or cryptographic mechanism, we used mechanisms that were developed to prevent overfitting, in a sense, that are in this particular case dropout and model stacking. So dropout is used essentially with neural networks. It consists of randomly removing edges in the neural networks. And model stacking is used for other machine learning models that we study, where it consists of instead of putting having different models, like in random forest, you have multiple decision tree, the same layer. Here we have multiple layers, actually. And the first layer contains some machine learning models, and then the output of this machine learning model are used as input to the next layer, where you have another machine learning model. And this has been shown to reduce overfitting. So let's look at the results. So recall and precision first. Okay, so we see the original attack in blue, original membership inference attack in blue, and you see the results with dropout or model stacking in green and red, respectively. And we see that, well, in general, the recall and precision drop by 20 percentage points for most of the data sets. Okay, and so this is the privacy gain that we have. Okay, but now let's look at how it affects utility, because we should also keep that in mind all the time, right? So now let's look at the accuracy of the original machine learning classifier that we should not forget, right? So here we have in blue against the original accuracy, and in green and red the accuracy with dropout and model stacking. And we see, in fact, that for some data sets, for about half of the data set, at least for dropout, the accuracy increases on the right-hand side for the purchase data set, for instance. And for the others, actually, the accuracy does not decrease much. So this is due to the fact that actually by preventing overfitting, you increase accuracy on and generalization of your machine learning model, right? And this, as such, this helps the utility overall, right? So we see with this particular example that privacy and utility are not always at odds with each other. In this particular case, with this particular attack, they can be both optimized at the same time. Okay, so to conclude this lecture, we have seen that we can leverage machine learning or statistical analysis methods for attacking privacy at scale. We have seen 3 different types of attacks against various types of biomedical data, but also this could apply to other types of data, such as location, as we have seen, but also hashtags. We have an example, also a paper on inferring location from hashtags using random forest. And we have, as I said, these 3 main types of attacks that are well established. In general, we have also seen how we can improve privacy in some machine learning settings. For instance, improve privacy with when we use support vector machine classifiers with MicroRNA expression, when we use random forest classifiers, or when we use machine learning as a service more generally. And this applies to many different machine learning models. But we see that in general, it's difficult to optimize both privacy, utility, and efficiency, right? There is no one-size-fits-all solutions. It depends a lot on the machine learning settings and the machine learning algorithm on one hand, but also on the machine learning phase, whether you are at the training phase or at the test phase. We have seen, for instance, for the private random forest classifier that we could only implement it and have some results in reasonable time at the test phase, at the inference phase. We have also seen that this depends highly on the attack success, what privacy protection you can use, and this depends also on the type of data and whether data is high dimensional, whether it's stable over time, and so on and so forth. But in general, it remains a grand challenge to optimize all these 3 dimensions: privacy, utility, and efficiency in the machine learning context. So if you have more questions, do not hesitate to contact me at this address. Or if you are interested in working with us at the Cyber Defense Campus, we have fellowships. We have also master projects. We have PhD projects through these fellowships or postdoctoral positions as well. And these are the references of the work that I presented today. So yeah. Thanks. I'm finishing a bit early, but feel free to ask any question now that we have a bit of time.
Thank you very, very much. And you have some time to write your questions in the Q&A, and I wrote my questions down, so I'm going to start with them. So the first one is by Camille Aradiani, who is asking, for the previous presentation, do you use specific libraries for security computation, like multi-party computation?
Which presentation which work are you referring to exactly?
I think this is about your second set.
The random forest, you mean?
Second set.
Right? Yes, so we didn't, as far as I can remember, we did not use any specific implementation. No, we just used like some, actually we built it on the work of Bost et al., which was published on NESS, which was actually doing this kind of using also additively homomorphic encryption for decision trees, and we extended it to random forest, in fact. But I'm not sure there were some libraries already available, or if we implemented it. But we used some like traditional schemes like Bayesian scheme.
I expect Camille to answer if he has further questions. My personal question is, have you looked at trying to reverse engineer biometrics? Quite a lot of the biometric systems these days are actually using machine learning to train, for example, face recognition. And have you tried to look at something like that? Or
to kind of to do like some kind of adversarial learning, or to reverse? To
reverse for the inference, to know whether you were in the training set, or, for example, if we discuss in a more global context,
yeah.
With a colleague of mine, we worked in the past on the old systems of machine learning used to have adversarial, not adversarial, the active appearance model, where you just compute at the end you project the face on different faces and you just check the linear combination. So for this one, it's very easy to reconstruct who were in the training set. The question here, have you looked at things which are more advanced?
No. Actually, I've never worked with biometric systems, but this would be a great application case. Actually, yeah, it's a nice idea, at least. Yeah.
And another question, and I see this from a lot of companies, they're saying, because biomedical data is very, very sensitive, so we're going to do the following thing. We're going to learn using the best learning tools on the raw data. Then we're going to use this machine learning classifier as a basis for simulating data. So they learn on the real data. Then, now, of course, you have something which is very sensitive because of inference attacks. And we're going I'm going to say we're going to switch it into a gun for the sake of argument. Of course, exact technique is less relevant. And then you press a button and you get medical records which are completely synthetic on one hand. On the other hand, they're supposed to carry the same amount of information.
Yeah.
Comes the question, from your perspective as someone attacking this system, what would you say?
I think there is actually a paper published this year at CCS that shows how much generative models GANs can leak about the membership inference in the original data set. I don't know if it's with biomedical data, probably not. It would be interesting to test with those, but probably with typical image data used in the setting, but yeah. So I don't know the results, whether this is possible, but this would be actually the attack that we can think about, right? To kind of infer whether, I'm not sure we can reconstruct the original data, to be honest. This would be another question, right? More challenging inference. But still inferring whether you were a member of this data set originally, provided your data is kind of already kind of a threat, privacy concern, right?
Yes, so the question, so your intuition, given the fact that you haven't done the research yet, or you're doing it at the moment and you don't want to tell us about it, which is completely fine, you should keep your privacy. The question is indeed, as an expert in these things, would you expect the answer to be, yes, there are going to be attacks, it's just a matter of putting the things in the right order, or, well, it seems like it's going to be significantly harder, but to the extent it's impossible to do that.
But it always also depends on the dimension, as I said, on the dimensionality of the data you have access to, and on the number of samples that were used originally, right? Or in the model, right? And the fewer samples were used in the model to construct the model, the more impact one sample has on the model, right? And if you have a lot of dimensions about this data, then you can probably run this attack quite efficiently, these membership inference attacks, even against this synthetically generated data, honestly. Yeah.
Are there any other questions? And I'm asking people to complain over the chat or the Q&A. It seems like everybody are either hungry or protect their privacy and their medical data. So, oh, here's a question. If I tend to keep my privacy, what should I do to protect it? Should I ensure that my data will never be published?
Yeah, as we have seen in the first part of the presentation, even if you protect your privacy well, your own data, sometimes your peers or your family members can reveal a lot about yourself, right? Through social networks, like in the Cambridge Analytica scandal, or by sharing their own data, like in genomic data, right? For genomic data, it's very stable over time, as I said. So it's very important to protect them well, to be sure that they are well protected with whoever you share this data with. For other types of biomedical data that are more variable over time, you might have a chance to preserve your privacy in the long term. But still, as I've shown in some cases with methylation data, you can link it back with genomic data, which then can de-anonymize yourself, right? So it's a tricky problem. Actually, these biomedical data are not well understood even by biomedical researchers at the moment. So it's even more complicated for privacy researchers like me. But there is a lot to do.
I'm guessing that you're not suggesting that I should go and start mass killing my family and extended family and that sort of stuff. and what about trying to drown the real data in fake data? I earlier asked about, a button which you press and simulates data, but let's assume for a second that I publish my record with multiple wrong readings.
Mm-hmm.
Would that be useful as a protection mechanism?
Yes, actually, because there is no check of the integrity of the real nature of the data for an adversary who has access to it. So except if the adversary is 23andMe or family tree DNA and they want to build a big database or share it with Google and do some kind of health insurance in the health insurance market, right? But these guys probably know they have the real data. And if you want to publish it online, then of course you could fake the data.
Regarding the MI attacks on the ML models, from observing and reading the relevant papers, it seems that a very high overfitting is required for the success of this attack. Or in some cases, some data set, this overfitting was manually created on purpose. So the question whether or not the membership inference attack is applicable in the real world setting.
Well, it depends what you mean by real world setup, actually. The setting is basically machine learning as a service. So, and in these models, the used data sets are image data sets and so on. So they are not, they're probably images, right? So like to CFAR and ImageNet and so on. So I don't know. For instance, we haven't tested actually the biomedical setup. If you build actually your model based on biomedical data or genomic data, which is now the case more and more, we haven't tested that, and actually this would be interesting to carry out. Because we have used the traditional ML data set like CFAR, like purchase data set, like MNIST, which are, indeed overfitting, right? Because the models are built on that essentially. But this would be interesting indeed to check, for instance, biomedical data. Yeah.
Okay, thank you very much. Are there any other questions from the audience? Ten, 9, 8. I'm trying to see. Okay. If If not, I would like to thank you very much for the talk. For all of the rest of you, we will reconvene at 2:40 Israel time, which is besides being in 50 minutes today, we're going to give Indian time zone for which is I think 2.5 hours difference. So it's going to be 12. Sorry, plus 2:30. Plus 2:30. I'm not going to do time zones today. Sorry, can't do the math. It's too complicated, and it will hurt the privacy of some of our participants. So see you in 53 minutes. Goodbye.
Bye.